Hello, and welcome to another episode of the JASN podcast series, a monthly podcast where we highlight an exciting paper from the current issue of the journal. I'm Alan Yu, Professor of Medicine at the University of Kansas Medical Center and Deputy Editor at JASN for Basic Sciences. Today, I'm joined by Dr. Chang from National Taiwan University Hospital in Taipei to discuss their exciting new paper entitled Cell-Specific Roles for Angiopoietin-2 in CKD, which appears in the current issue of JASN. So welcome, Fanchi. And to start off with, please can you introduce yourself and tell me a little bit about your background?
Thank you. Hi, everyone, and also, of course, Professor Yu. It's a pleasure for me to be here. I'm Chang. I'm a nephrologist and physician scientist at National Taiwan University Hospital in Taiwan. And Taiwan may be best known for our internationally well-known semiconductor industry, but in the nephrology field, we are also known for having one of the highest rates of kidney failure in the world. So CKD is pretty much a frontline problem for us. And clinically, I care for patients with kidney disease of all kinds, and of course, especially chronic kidney disease. And on the research side, my team has spent many years trying to understand why these tiny blood vessels inside the kidney deteriorate as kidney disease progresses, and what we can do to protect them. So that question led us to the angiopoietin family, particularly to angiopoietin-2, which keeps coming up as a key driver of vascular injury and, of course, kidney scarring. We've been following that thread across multiple studies over the past decade, and this new exciting JASN paper is the latest chapter.
Thank you so much, Fanchi. It is indeed exciting. So this family of proteins that you call angiopoietins, I'm going to simplify it, and most of our readers, of course, are familiar with erythropoietin, and based on the name, I think they can surmise that angiopoietin is a growth factor as well, but it's one for the vasculature. And perhaps the readers are more familiar with VEGF as a growth factor for vasculature, vascular endothelial growth factor, which acts on VEGF receptor and stimulates proliferation of new vessels. Angiopoietins, as I understand it, work through their receptor Ty1, but instead of stimulating new vessels, it's sort of maintenance, right? It regulates blood vessel integrity and stability. And simplistically, from my reading, angiopoietin-1 promotes stability and integrity, while angiopoietin-2 disrupts vessel stability and makes them leaky. So correct me if I'm wrong on that, and perhaps you could walk me through how you came to study angiopoietin-2 and CKD, and what your both previous studies and some of the initial studies in this paper showed about the relationship between angiopoietin-2 and human CKD and outcomes.
Yes, you already explained clearly about the basic function of angiopoietins and their receptor type 2, their main receptor. The initial spark of our research came from our clinical observations. When we measured angiopoietin-2 levels in the blood of our CKD patients, we found that they rose progressively as kidney function declined and correlated with protein in the urine, which is albuminuria, and with signs and markers of inflammation. That was published back in 2013. The time we don't have SGLT2 inhibitor, GLP-1 receptor antagonist, or non-steroidal miracoglia receptor antagonist. The best we can do for our CKD patients are basically supportive care, lifestyle modification, and RAS inhibitor. So we then followed 319 CKD patients for several years, and we found that high angiopoietin-2 levels were independently associated with the risk of reaching kidney failure or death, even after accounting for traditional or standard risk factors. And we published a 2014 JASN paper linking high angiopoietin-2 to arterial stiffness connecting angiopoietin-2 not just to kidney scarring, but to cardiovascular complications as well. But that was all based on actually blood levels. What's new in this JASN paper is that we actually look directly at the kidney tissue. We use RNA sequencing. Essentially, we read the gene activity from kidney biopsies of 127 CKD patients in a prospective cohort we run at Taipei Veteran General Hospital. And what we found was actually striking. The higher the angiopoietin-2 gene expression in the kidney tissue itself, the lower the kidney function, the greater the degree of fibrosis on biopsy, and the higher risk of reaching kidney failure or death. Importantly, angiopoietin-1, the type 2 agonist, showed none of these associations. So it was angiopoietin-2 specifically tracking with disease severity and progression. And this is the first time anyone linked kidney tissue angiopoietin-2 levels to clinical outcome in this way. And actually, on the experimental side, we know from earlier work that angiopoietin-2 is normally very low in healthy kidneys. But in mouse models of progressive kidney disease or fibrosis, it gets really upregulated, particularly in the kidney tubule cells, while angiopoietin-1 goes down. So this imbalance, too much angiopoietin-2 to little angiopoietin-1, actually became a recurring theme across our studies.
So angiopoietin-2 not only an independent biomarker of CKD severity and progression, but it seems like it's actually being made in the kidney and upregulated in disease. So the obvious next question is, so does the angiopoietin-2 actually play a causal role in the CKD progression, or is it a compensatory response that might have a protective role? And so that's kind of the crux of your paper, right? And how did you distinguish this experimentally?
Yeah, this is the classic chicken and egg question when we just talk about observational data. Yeah, that everyone will raise this issue. Is angiopoietin-2 driving the disease, or is angiopoietin-2 is just a bystander being released by a damaged kidney? And like what you just mentioned, I will add, even if it is causal, or can we actually modify it to make a difference? So to answer both questions, we try and turn to experimental mouse models. In our 2022 KI paper, we showed that genetically boosting angiopoietin-1 in kidney tubule cells, which naturally counters angiopoietin-2, reduced macrophage infiltration, protects the capillaries, and also reduce fibrosis. And when we directly blocked angiopoietin-2 using a pharmacologic inhibitor, we saw the same protective effects. So actually, these two interventional experiments converged on the same answer. Angiopoietin-2 is not just a marker. It is actively driving disease. And in this new JASN paper, we went a step further. We generated mice with angiopoietin-2 completely deleted. Essentially, we turn off this protein altogether. And then we induce kidney disease with an adenine-containing diet, which is a CKD model with abnormal kidney function. These mice were significantly protected. They have better kidney function, less inflammation, fewer capillary loss, and of course, less fibrosis. And importantly, deleting angiopoietin-2 in otherwise healthy adult mice had no adverse effects, which is very reassuring from a safety standpoint and really supports angiopoietin-2 as a viable therapeutic target.
Yeah, thank you. I think that was really the key and most interesting finding in your paper. So, of course, the next step is to try and figure out mechanistically how angiopoietin was working and I guess what cells were responsible. And I assumed, as most people would, that angiopoietin-2 must be expressed in the vasculature. But here's the spoiler alert, turns out it's not that simple. So tell us what you found.
Yes, this is where this story gets really interesting. Most people, like what you just said, that will assume Angiopoietin-2 is simply made by the blood vessel lining cells, which are endothelial cells, and acts on those same cells, the Tie2 receptor of endothelial cells. But here's the thing. As kidney scar, those endothelial cells actually start to die off. They start to ratify, they start to disappear. So if they are disappearing, where is all this angiopoietin-2 coming from? And we found that in damaged kidneys, the kidney tubule cells, the cells responsible for filtering and reabsorption, become a major source of angiopoietin-2 overproduction. So it is the tubules making this protein and send it over to the nearby capillary cells to act on them. And here's the really novel finding of our paper. We engineered mice that lacked angiopoietin-2 specifically in endothelial cells, and separately, mice that lack it only in tubule cells. These two experiments told very different stories. When we knock out angiopoietin-2 just in endothelial cells, it suppressed early inflammation nicely with fewer inflammatory signals, fewer macrophages coming in at the start, but it did not prevent the late-stage scarring or capillary loss. But by contrast, knocking it out specifically in the tubule cells did not affect early inflammation much at all. But it dramatically protected kidney function and reduced fibrosis in the advanced stages. So what this tells us is that angiopoietin-2 plays different roles at different stages of kidney disease, depending on which cell type is producing it. Endothelial-derived angiopoietin-2 drives the early inflammatory fire, and tubule-derived angiopoietin-2 sustains the later destructive scarring. So that is the cell-specific, stage-specific story that gives our paper its title.
Yeah, thank you. So the effect of the angiopoietin-2, you think it's dependent on the timing of when it's secreted, or it's dependent on which compartment it's released from? A bit of both.
Yeah, I think both contribute, and I think, of course, that's another thing. I think is the injury model. Like if this injury is particularly driven by endothelial cell in certain kidney disease, then of course endothelial-derived angiopoietin-2 may play a bigger and major role throughout the disease course. But for CKD is the endpoint of different kind of kidney disease. Yeah, so at the later stage, it's totally different.
Very interesting. I hope at some point you'll take a look at my favorite kidney disease, which is polycystic kidney disease, where you may be aware that vessel rarefaction is a quite striking early finding. So, but I digress. Let's finish off by talking about the translational implications, which you already alluded to. So do you think that inhibiting angiopoietin-2 is a viable strategy for treating or preventing CKD? What about upregulating angiopoietin-1, which seems to be antagonistic to angiopoietin-2? Yeah. What would be the effect of activating the receptor Tie2? And which of these are most feasible or the most logical approach?
This is the most exciting part, and honestly, it's where I think our findings have the most direct clinical relevance. And the biology, of course, points to several potential strategies, like you just mentioned that, but they are not all equally promising, I think. So let me just try to walk through them. The first one, upregulating angiopoietin-1, is conceptually appealing, but here's the nuance. Angiopoietin-1 administration alone may or can paradoxically worsen inflammation and fibrosis in certain injury settings. This has been shown by some groups. So it may not be as clean a target. And another strategy is to activate Tie2 receptors directly, essentially bypassing the angiopoietin imbalance altogether. And a recent 2025 study used a smart engineered antibody that simultaneously blocks angiopoietin-2 and also activates Tie2. And it shows strong protection against capillary loss in preclinical models. This dual action is elegant in concept, but I do want to flag two practical points, also concerns. First one is that as CKD progresses and capillaries are lost, there are simply fewer endothelial cells left that carry the type 2 receptor. So the target itself may be diminishing just when you need it most. And the second one is that type 2 receptor is expressed broadly throughout the body's vasculature, not just the kidney. So long-term activation carries the theoretical risk of unintended vascular side effects. So these are not deal breakers, but they do mean that we need to think carefully about how to modulate this pathway safely. So this is why I think direct angiopoietin-2 inhibition is our most promising strategy. The biology is very compelling from a safety standpoint. Angiopoietin-2 is present at very low levels in healthy kidneys, but gets strongly upregulated, specifically when disease kicks in. That disease selective profile means we are targeting something that's driving pathology without disrupting normal vascular homeostasis. And we have validated this in our preclinical models. And there is already a clinical signal pointing the same direction. I read a paper that's a post-hoc analysis of the Credence trial. They found that canagliflozin, the SGLT2 inhibitor, significantly lower angiopoietin-2 levels. That's also a suggestion that part of its kidney protective effect may actually run through this pathway. So to answer your question directly, yes, I do think angiopoietin-2 inhibition is a genuine therapeutic strategy for CKD. It's not just a biomarker story. And the next step has to be a well-designed clinical trial. And I do hope our work helps make the case for why that trial is worth doing. And of course, as with any targeted therapy, an important question going forward will be how to identify which patients are most likely to benefit. That is something the field will need to think about as this moves toward the clinic.
Thank you so much, Fangchi. So are you, or is there any company you're aware of that's developing an anti-angiopoietin-2 molecule of some sort?
There were some targeting this pathway, anti-type 2, anti-angiopoietin-1, angiopoietin-2, or specifically to angiopoietin-2 monoclonal antibody in cancer field.
I see.
Yeah, as an adjuvant therapy, but the result of that clinical trial, they didn't look good. And for kidney field, actually, there are no designed drugs for this. But I think this is a good way, especially nowadays we have different kind of new tools, not just for monoclonal antibody. We have maybe small molecule inhibitor. Yes, that is a good thing.
Thank you so much. Well, that is pretty exciting, and I'm looking forward to see how this field unfolds. Again, thank you, Dr. Chang, for publishing your paper in JASN and for chatting with us today.
Yeah, it's our pleasure to share the findings with all of you. Thank you.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.